So our last talk today, and we'll finish this with a short summary by Ray Zimmerman after our last technical talk today, is by Eugenia Naro-Maciel. And this will probably be a fairly new topic for some of you in terms of environmental DNA. And I think it's nice to bring in something alive, some biology and some concepts of living organisms. We tend to think about cities as kind of places of concrete, glass, and steel. But when we look around us, particularly cities like New York, they're actually filled with plants and trees and birds and squirrels and all sorts of interesting things. And those are just the things that we can easily see. So I'm going to turn this over to our final technical speaker. Thank you.
Thank you. Hi, good afternoon. Everybody can hear me okay? So it's a great pleasure to be here. I work at NYU Liberal Studies, and it's a very interdisciplinary program. But this, to me, is also very interdisciplinary, working with engineers. And I think it's just a wonderful idea. I want to thank Deborah and Brittany for organizing such a great space where we can all kind of share different ideas. And so I'm going to talk a little bit about environmental DNA. But before that, I'm going to just discuss the kind of the global biodiversity crisis that we're in, and also how next generation technology in genetics can help provide information about that. And then I'm going to come into one of my favorite studies right here in New York City about oysters and monitoring oyster reefs. And so I know that it's the end of the day and people might be a little tired, so I thought I might ask you just to talk to your neighbor or the person behind you just very briefly about what do you know or not know. Have you ever heard of these ideas that we might be in a sixth mass extinction event, or that we might be in a whole new geologic time period called the Anthropocene? So I just want to encourage you to take a second, and maybe you'll meet somebody new and just talk about these questions very quickly. So I wasn't expecting that to go so well. I get the sense that you guys are interested in this stuff, so that's great. So did we get a consensus? Are we in the sixth mass extinction? Had anyone thought about that? And are we in a new time period? So these questions are still open for debate. And I guess, if you want to know what I think, I would say we still have time to not get to the sixth mass extinction level. If we keep going at this rate, we might get to 75% or more loss of species, which has only happened 5 other times in the history of life. In the past 500 million years or so. And I do think that we're in the Anthropocene. So I think we've changed the world enough that future layers of fossils will be different than what they were before. But there's no doubt whatsoever about how urgent the situation is, right? So we've had this year these 2 major reports that came from UN bodies, and I think probably many of you are familiar with them. There was the IPCC report on the climate change panel saying that we need to not get above 1.5 degrees Celsius increase above pre-industrial levels, and that is very important in terms of human well-being as well as wildlife. And then we had this other report which had a sea turtle on the cover, and that happens to be my field. So I was very happy to see the sea turtle, but I wasn't happy to read about, about 1 million species are expected to be in danger of extinction, and these 2 reports are not based on just an opinion. They're based on very, very solid science. They're extremely well supported. They've been looked at by scientists all over the world. And it's also a good sign, though, because, and that's why I like coming to give talks at places like this where maybe people aren't necessarily, focused on this as much, because it's a time where we have this, very young people, even just 16-year-olds, coming out and saying, this particular young lady, Greta Thunberg, has said, in her opinion, we shouldn't be talking about anything else. We should only be talking about this because of the vast repercussions for people and for wildlife. And so it's a good time. And with engineers, there's a lot that engineers could be doing and are doing to help with this. And what am I doing in terms of being a biologist? And one of the things, of course, I'm doing is teaching. But one of the areas that I'm very interested in, it's really undergone a tremendous revolution just in the time that I've been working, is that we have these next generation genetic approaches. And so when we talk about biodiversity loss, sometimes we don't even know what we're losing. Like we may know about whales and sea turtles and organisms that are very big or sexy or that people love to study, but what about krill and plankton and things that are the basis of the food chain? We might not know very much about what's being lost here. And usually it's very labor intensive to try to find out what kind of organisms are in the system, especially if they're microscopic. And you need a lot of expertise and studies. But DNA is providing a good complement to that, where you can identify organisms from their genetic sequences. And that's called DNA barcoding. And then with this revolution, now, it took me, for example, when I was in college doing my senior thesis, a year to get 100 sequences. Now, in a couple of days, you can get millions and millions of sequences. So this field is just really booming, high-tech field. And so this is just a regular DNA barcoding where you have one sample of a sea turtle or something like that. You sequence that organism. You get a DNA sequence. You can put it on an online free database, and it'll tell you what it is. So, for example, there was a study done by Natural History Museum scientists of caviar, where they went out and bought a bunch of caviar all over the city, and then they sequenced the caviar and they submitted it to this database, and they found out that a lot of the caviar was mislabeled, and that people were paying a lot of money for something that wasn't really what it said on the can. There was also some properly labeled caviar. And then this is this exciting technology that I've become involved in, where you can just get a liter of water or a sediment sample, or if you really wanted to, you could do a scraping off a subway platform, which has been done, and obtain from that millions of sequences. So millions and millions of sequences. And so, that's really helping us understand what life on Earth is. Well, we sometimes think of life on Earth in terms of people, but actually most of life on Earth in terms of diversity is bacteria, right? And we didn't know that before because we couldn't sequence it, we couldn't see it, and now we can. And we're talking here about cities, and I love New York City. I'm from New York City, and I want to research this city. And this is also one of the cities that is going to potentially be in trouble with climate change by the time we get to 2050 or 2100. And we're already seeing signs of it now with sea level rise, for example. And so that's why we're already got a plan in New York City investing billions of dollars to do engineering things, right? And build berms and keep the sea out. But there's all these other solutions. For example, when we had Hurricane Sandy, if we had had more oyster reefs and wetlands, the storm surge would have been mitigated. The oyster reefs would have blocked the storm surge, and the wetlands would have helped absorb it. And so there's this big effort to reinstall oyster reefs into New York. And this is the Bronx River. And this is an effort led by Allison Mass-Fitzgerald, and she's in Baykeeper. And there's also work with the Billion Oyster Project. And what they do, this is, you can see, labor-intensive morphological monitoring. They'll go out and spend a lot of hours measuring oysters to see how they're doing in this, the Bronx River. You might think they might not be doing very well there, but actually they are doing fairly well. And so what I did was to help out her team, and my student, Irina Werner, led this, And we wanted to see what else was growing in those waters along with the oyster reefs. And so we did go into the Bronx River. I don't know if I would recommend that, but we did find that we found millions of sequences, right? And many things that people didn't know hadn't really identified there because they're microscopic plankton. And we found that the communities in the water was different from what was in the sediment. Right? We had more of these plankton in the water, more worms and nematodes, things like that in the sediment. We saw the waters were all mixing, so they had sort of the same community in the water, but the sediments in different areas actually seemed to be significantly different from each other, so different communities. And we were able to identify oysters. So we saw the oysters, we knew they were there, but we didn't know if we'd be able to pick their DNA up, and we did. And we were also able to find a pathogen that affects oysters that is very low frequency and sort of rare in this area, but we still found it. And so we know that that could be an early warning system. And so to wrap it up, we found that this environmental DNA technology is a great way to explore life under the surface. And I think it fits in very well with the emerging technologies here. Thank you.
Questions for Eugenia?
I think these DNA-based assays are really useful for identifying things that are similar to things that you already know. But does your project also attempt to assemble novel sequences, novel genomes?
Yeah, so that's a great question because these methods are only as good as the reference databases. So it's possible, for example, I can ask the database, what is this, microscopic plankton? And it'll say, well, I don't know what that is. I've never seen that before. I can tell you what the most similar thing to it is. And that's where we're starting to see really how much that we don't know. So these are a starting point to then go on and name species and learn about them with morphology also. So sometimes we use operational taxonomic units where we say, we don't know what this is, but this is an operational taxonomic unit for now.